Bismillahirrahmanirrahim. Let's talk about how different biosensors can be used to find out toxicity of different samples and different chemical compounds. There have been much advancement in this area. Now, what mostly we do is that we do the chemical analysis and use different analytical machines to find out the concentrations and the presence or absence of different toxicants in certain samples. When we do this kind of analysis, this does not give us sufficient information about the ecological risks of that toxic compound or the sample. वो से हमें ये बताता है कि इसमें कितनी कंसंट्रेशन है, लेकिन उसके जो इफेक्ट्स हैं इकोलॉजी पे या लाइफ पे वो ऑब्वियसली एज सच वो नहीं बता सकता unless until we put some experiment or we find it out at the site. Now much efforts have been done to develop and use different bioassays and biosensors to find out the toxicity of different water samples and the effect on different life forms. तो biosensors का एक एडवांटेज ये भी है कि ऑन साइट आपका डिटर्मिनेशन हो जाती है कि कोई कंपाउंड प्रेजेंस है या एब्सेंस है, प्लस उसके इकोलॉजिकल इफेक्ट्स वेदर दोज आर हार्मफुल और नॉट वो भी सामने आ जाते हैं. सो दैट्स व्हाई biosensors have their own importance. There have been different types of biosensors जिनका हमें लास्ट लेसन में डिस्कस किया था. For example, whole organisms can be used to measure the potential biological toxicity of different samples. जिसमें हमने बात की थी कि whole cells biosensors are there as well. There have been different types of toxicity assays which are based on this approach. For example, microtox and tox alert are there, which have been widely used. These are basically based on the use of luminescent bacteria, Vibrio fischeri, to measure toxicity from samples. Based on the luminance from the bacteria, we can tell whether some compound is present or absent, or whether some compound is toxic or not, depending on the metabolic processes ongoing in the whole bacterial cell. The luminance produced by the cell will be more or less. Cell sense is another type of bioassay. Basically, it is an amperometric sensor. It incorporates Escherichia coli cells, and it is used for ecotoxicity analysis. Again, if we want to find out whether some chemical compound is toxic towards different ecosystems or not. Basically, it uses ferricyanide, which is a soluble electron mediator. Electron को mediate move करने के लिए use होता है. So basically, it diverts electrons from the respiratory system of the immobilized bacteria, E. coli basically, and it moves it to electrodes. So due to this movement of electrons, a current will be generated. The more the current is generated, it means the more the respiratory process is going on in the cell, which means the cell is in much better health and the compound is not toxic for the cell. However, if there are any changes in this current generation, it means that the respiratory processes of the immobilized bacteria are not going on so well. So it means the compound which has been exposed with that biosensor, the compound is most likely to be a toxic compound. So such biosensors have been used to find out the toxicity of so many different compounds. For example, 3,5-dichlorophenol and other phenols, for the determination of non-ionic surfactants, for benzsulfonate compounds, and for the analysis of wastewater treatment processes as well. Most of the environmental biosensors which are used are basically based on bacterial systems, either whole cell or the enzymes and proteins derived from the bacteria. Eukaryotic biosensors obviously are much rare, and even rare are the mammalian biosensors, the sensors based on mammalian cells or the proteins of the proteins and enzymes of the mammalian cells. However, if we use biosensors based on mammalian cells, it can give us much more information about the toxicity of certain compounds. Why so? Because if we expose a bacterial cell with a toxic compound, and if at the same time if we expose a mammalian cell with the same toxic compound with the same concentration, the responses can be different. A mammalian cell can behave almost entirely differently towards a certain toxic compound as compared to the response of a bacterial cell. If we see overall, the human body can have so many different complex responses when exposed to different toxicants. So that's why if a biosensor is based on mammalian cell, that can give us much more sensitive information and much more information about the response towards certain toxicant. This was how biosensors can be used to find out, determine the toxicity of different compounds in samples. We will talk more about biosensors in our next lessons.